So today's lecture is going to be about chapter number 4, enzymes and biological reactions. So the chapter is really important because this chapter has been important in the Cambridge era and the MUR era. In both of these eras, like before 2023 or after 2023, this chapter has been really important, and there has been a definite question in IMAT from this chapter. If we talk about the yield analysis, you can see that enzyme structure had like really high yield, and enzyme kinetics have also really high yield. And then you have comparative inhibition, and then cofactors, activation energy, types of reactions, and then ATP as energy currency. So these are the topics that are really important. You have to know that this chapter is not simply this chapter, but this chapter would be really important when it is coupled the concepts with bioenergetics and other chapters that are related to enzymes. Like all the reactions in other chapters would be discussed and like combined the concept with this chapter. So this chapter's concept is going to be the base for future chapters that you will discuss, like respiration, digestion and genetics. Those chapters will unlock more concepts for enzymes, but you have to make your base concepts in this chapter, and then you will move on to those chapters that are really important. The first thing there is that take a screenshot of this high yield concept list. So you need to know like what are the things that you need to study and what are the things that you don't need to study. So these are the topics that are really important for your IMAT exam. Then let's move on to our first thing, that is about what is an enzyme. So enzymes are like globular protein. The first thing that you need to know about is that enzymes are made up of proteins and they act as a catalyst. What does a catalyst mean? Catalyst is something that speeds up a chemical reaction. For example, when you eat food and your stomach start digesting it, it takes a few hours to digest the food. But if there are no enzymes in your stomach, then it will take a number of days or even weeks to digest a single piece of bread in your stomach. That's why enzymes are so important, because they speed the chemical reactions a lot. And the key principle is that they don't only speed up the reaction, they actually lowers the activation energy. What is activation energy and how it is lowered, and how does enzyme speed up reaction? We will discuss it in detail in our activation energy topic. So first thing that you need to know is that their presence does not affect the nature or properties of the reaction. Nature and properties, they remains the same, like reactions nature and properties not changes. So the reactions remain the same, only the thing that changes is that the enzyme actually speeds up the reaction. The nature and properties of the products and the reactants does not actually change. And the other thing is that they are used in really small quantity. Like you don't need a lot amount of enzymes to start or speed up a reaction. You can do that with a small quantity of enzymes. And the other thing is that they are really specific. You need to know that enzymes are highly specific. They only act on specific products and specific reactants. For example, you will discuss it in your examples about induced fit model and log and key model. Just like a key is fit into a log, that's how enzyme will also fit into a specific substrate, and that's how enzymes act. That's why they are really specific. They don't just react with any molecule, they are for specific molecules, and they only react with those molecules. The other thing is that you already know they are low activation energy, and the other thing is that they require aqueous medium. Yes, enzymes don't work in normal conditions, like they require aqueous medium. Why do they require aqueous medium? The reason is that because enzymes cannot show movement on their own. They don't have arms, they don't have legs. So how do they reach substrates? They will actually swim towards the substrates. Like the substrate is at point A and the enzyme is at point B. How will enzyme will go from point A to point B? Obviously, if they are in liquid or in aqueous medium, the enzyme will float towards the substrate. So that was about the aqueous medium. And the other thing is that they can also work in vitro and in vivo. They can work in both, like they can work in vivo and in vitro, both conditions. And the other thing is that like some enzymes are potentially damaging, like if they are activated in active form, they are really damaging, just like pepsin. Pepsin is really damaging enzyme, like it can damage your stomach if it is activated or released in activated form. Some are damaging, e.g. pepsin. Pepsin is actually an enzyme that is present in your stomach. Similarly HCl, both of these are combined together and they work together to digest food. And pepsin and HCl are so acidic that they can make a hole at your table. Like if you drop a few drops of pepsin and HCl on your table, then they can make a hole in your table, in your wooden table. So they are so dangerous, and these enzymes are present in your stomach, and if they are released in activated form, then they can potentially damage your stomach, and that's what is called as ulcer, right? Ulcer is nothing, just a hole in your stomach because of pepsin and HCl, if they are released in activated form. So that's why these enzymes are released in unactivated form, and unactivated form will slowly converts into activated form. So these were some properties that you needed to know about enzymes. The other thing is about the structure of enzyme. So we just discussed that enzymes are made up of globular protein, while there is an exception too, not all enzymes are made up of protein. So the exception is ribozyme. Ribozyme is the only enzyme that is not made up of proteins. Ribozymes, these are the only enzymes that are not made up of protein. So ribozymes are enzymes that are actually made up of RNA molecules, and they are found in ribosomes. Yes, these are the only enzymes that are made up of RNA, and they are not made up of protein, and they are found in ribosome. For example, the example is peptidal transferases. Peptidal transferase. This is an enzyme. It is actually a ribozyme which forms peptide bonds during translation. What is its function? It forms peptide bonds. Peptide bonds are formed by this enzyme during translation. Translation is the process in which proteins are made. So we will discuss it in detail in our next lectures. So peptidal transferase is an example of ribozyme. The ribozymes are the only enzymes that are not made up of protein, and they are made up of RNA. So now you know that what are enzymes and what kind of structure there could be, and what are the exceptions. Next thing is that what is the shape of enzyme. As you can see, enzyme has two structures. The first is the active site, and the active site is the site at which the substrate will come and attach. At the active site, if this is a substrate, then the substrate will come and attach at the active site, and the product will be formed just like this. This is substrate and this is a product, and active site is the site where the whole process occurs in an enzyme. Even the active site contains two sites. The first is the binding site and the other is the active site. So what is binding site? Binding site is a site that actually make bonds with the substrate, and thus it is involved in recognition and forming enzyme substrate complex, while the catalytic site is involved in the transformation of substrate enzyme substrate complex into a product. If we write them down, what does binding site and catalytic site do? So as we discussed right now, what does the binding site do? It helps in recognition, if the substrate is real or not, if it is the substrates that we need or not. And then binding site will form enzyme and substrate complex. It is called ES complex. ES complex is when enzyme and substrate are connected with each other, then if they will form enzyme substrate complex. And if we talk about catalytic site, as you can see, binding site helped enzyme and substrate to connect with each other and a complex is formed, while the catalytic site has the next step. Catalytic site will convert the ES complex, like ES complex would be converted into the product, like product would be formed by the catalytic site. So that's how enzyme works, and this is the active site. The other site that is other than active site is called allosteric site. What is it called? It is also called as allosteric site. The site other than active site is called allosteric site. So allosteric site, as you can see, is the site where the substrate will not attach. What is the function of allosteric site? Allosteric site actually gives enzyme the structure. Enzyme structure is made of allosteric site. So this was about the basic structure. As you can see, enzyme has in front of you, it contains active site. Active site has two parts, binding site and catalytic site, and then the site other than the active site, the whole surface, as you can see, is actually the allosteric site. It is protein structure. So let's move on to our next thing, that is about cofactors. So cofactors is a really important thing. If we talk about enzymes, then enzymes contains two kind of parts. What are these parts? Either enzyme could be a protein part or a non-protein part. Either an enzyme could be protein or non-protein. We just discussed that enzyme is made up of protein, but enzyme also contains non-protein parts. I'm not joking right now, okay? Enzymes are definitely made up of proteins, no doubt about it, but it also contains a small non-protein parts, and those non-protein parts are actually called as the cofactors. What are they called? They are called cofactors. So cofactors is the part of enzymes that are non-proteins. So what are cofactors? So cofactors is like non-protein part, and that is required for the proper functioning of enzyme, like that they are essential for enzyme. So what are their functions? First is that they act as bridge. So it is kind of a bridge between enzyme and substrate. The other thing is that it also acts as a source of energy. That's why the energy comes from the cofactor part, and it also helps to drive reaction which otherwise would be difficult, or you can say it would be impossible without the cofactors. So now you know, like the non-protein part is cofactor, and the cofactor has more types too. Like cofactor, the table does not end here. The cofactor has actually two types. If we talk about cofactor has two types. Cofactor either could be inorganic or either it could be organic. So the non-protein part now has two types. The protein part could be, non-protein part could be organic or inorganic. If we say that if the part is inorganic, then the inorganic part is actually called as activator. So the inorganic part is called activator. What is an activator? So the inorganic factor that is attached to enzyme, like they attach or bind with the substrates, and they help to activate the process. They are actually detachable. They are not like, you know, physically bonded with the enzyme. That's why they are called activators. The examples are like ions. Ions are the best examples of activators. What are the examples? You have like studied about these kind of enzymes a lot, just like iron, magnesium, and copper and zinc. These are all the examples of activators. If we talk about organic part, then organic part contains two types. Like the organic part could either be coenzymes, or either it could be prosthetic group. Now the organic has more two parts like coenzyme and prosthetic group. If we talk about first of all are coenzymes, so if the cofactor is organic and if it is loosely attached, the first property of coenzyme is that it is loosely attached with the enzyme. So the non-protein organic part that is loosely attached with the enzyme is called as coenzyme. The first thing that you studied about coenzyme is that it is loosely attached. The next thing is that coenzymes are actually derivatives of enzymes. That is the other property of coenzyme. They are derivatives of vitamins. For example, what are the examples of coenzymes? For the examples are e.g. you studied about these derivatives already. They are ATP, then you have NAD positive, and then you have FAD positive. So these are all the examples of coenzymes, and the coenzymes are like really important because you know that ATP, NAD and FAD are so important in your respiration chapter that they are like present in every single reaction. If we talk about prosthetic group, if it is covalently attached, like coenzyme was loosely attached but prosthetic group is covalently attached with the enzyme, then it is called as prosthetic group. It is like permanently attached to the enzyme and does not detach even after the reaction. So the example is like, you know, e.g. the example is iron that is containing porphyrin, iron containing porphyrin ring. So the iron containing porphyrin ring is actually attached to some enzymes like cytochrome. I think we will remove the whole part and write it again. The example is iron containing porphyrin ring that is actually attached to enzymes like cytochrome. We will discuss cytochrome and porphyrin ring in our chapter that is actually photosynthesis. In the photosynthesis chapter we will discuss about iron containing porphyrin and cytochromes. So this is the example of prosthetic group. So we discussed about coenzymes, activators and prosthetic group. If we revise all that stuff, you know that enzyme contains two parts, protein and non-protein part. Non-protein part could be cofactor, and the cofactor is actually the bridge between electron and substrates, enzymes and substrate, and they are actually the source of energy. The cofactor could be organic or inorganic. If it is inorganic, then it is called as activator, and if it is organic, then it is called coenzyme. If we talk about inorganic part, the activator, the example is ions. The examples are iron, magnesium, copper, zinc. They are the examples of for activators. If we talk about coenzymes, then if they are if the organic is loosely attached, then it is coenzyme. If the organic part is covalently attached, then it is prosthetic group. The example of coenzymes are simply, we just discussed them. They are ATP, NAD and FAD positive. If we talk about prosthetic group, then the example of prosthetic group is iron containing porphyrin ring that is permanently attached to the surface of cytochrome, and these are the proteins and non-protein parts of enzymes. We discussed them in detail, and if you still have any question, you can ask me after the lecture. The next thing is that there are also two types of enzymes. What are the two types of enzymes? They are actually Holoenzyme and Apoenzyme. Let me write it down here. What are Holoenzyme and what are Apoenzyme? So first of all, if we talk about Holoenzymes, and then you have Apoenzyme. They are really easy. You don't need to be really confused about these names. First of all, if we talk about holoenzyme, then holoenzyme means that the enzyme is complete, it is whole. If then activated enzyme consists of a polypeptide chain plus it contains a cofactor, then it is a holoenzyme. It is whole, like it contains the polypeptide chain and also contains the non-protein part, that is the cofactor. But if an enzyme contains with its coenzyme has been removed, or you can say it only contains polypeptide chains, only the protein part, but no cofactor, then it is called as apoenzyme. Like the coenzyme or the prosthetic group has been removed, then the enzyme would be called as apoenzyme. So there is holoenzyme and then you have apoenzyme. So these are two types of enzymes that you will study in this chapter. So holoenzyme contains cofactor and apoenzyme does not contain cofactor. Holoenzyme is whole and apoenzyme is like half, or you can say incomplete enzyme. So this was about the introduction of enzyme. If we study about the processes, or you can say mechanism of enzyme action, the first thing that you need to know is that there are two models to understand the mechanism of enzyme action. So how does enzyme forms a chemical reaction? So first of all, you will have an enzyme and then enzyme will combine with a substrate, right? And then after combining with substrate, it will form enzyme substrate complex. Easy, right? It's not that difficult. Then after the enzyme substrate complex, you will have enzyme product complex, like the substrate would be converted into product. After this complex, then you will have different enzyme and products, like enzymes and product would be released. So that's how you can say the enzyme action actually works. But there are two models that explain this whole process. The first is the lock and key model, the other is induced fit model. So first let's discuss about lock and key model. So lock and key model was actually given by Emil Fischer in 1894. So according to this model, the active site of enzyme has definite shape, or you can say it has a rigid shape or structure, and there is no modification or no flexibility in the structure in the active site of the enzyme during or after the enzyme action. The enzyme which work according to this model are called as regulatory enzymes. So the enzymes that follow this model are called regulatory enzymes. The next thing is that you need to know about lock and key model is that enzyme act like a key and substrate act like a lock. Just like a specific key unlocks a specific lock, you cannot open a lock without any key. Similarly, a specific substrate is opened by a specific key, that is the enzyme. So enzyme act as a key and the substrate act as a lock. So the example of these are the enzymes just like sucrase, maltase. Sucrase only acts on sucrose while maltase only acts on maltose. So these are example of lock and key model. If we write down the examples, these are sucrase and then you have maltase. These are really common examples of lock and key model because sucrose only digests or acts on sucrose and maltase only act on maltose. So this was about lock and key model. So lock and key model tells you that when one enzyme can catalyze only one substrate and essentially no other enzyme could be catalyzed, and it is also called as like the absolute specificity, like enzyme is absolutely specific to the substrate, like a specific lock can only be opened by a specific key enzyme. The other model that was given by Daniel Koshland, and this model tells you that the enzyme is like not rigid, not non-flexible, but the active site of the enzyme is actually flexible. The active site of enzyme can change. As you can see in the above diagram here, the active site fitted the substrate exactly just like they were. If the substrates were round or if the substrate was in the star shape, then they wouldn't fit into the active site. But in induced fit model, they are saying that if the shape of substrate is different, then the enzyme will change its active site. As you can see, there were two triangles, but the substrate had a shape of a square, but the active site of the enzyme changed from triangle to a square so that the substrate could fit. So as you can see, the active site in this process is a bit flexible. Induced fit model is telling you that active site is not really rigid, but active site is actually flexible. It can be modified as the substrate interacts with the enzyme and the reaction is completed. The active site remains or regains its original shape after the reaction. So this is the flexibility of active site, which allows more than one type of related substrate to be attached on active site, and that's why an enzyme can carry out different chemical reactions. So the example of this are carbonic anhydrase. Carbonic anhydrase is like a really common example for this reaction. Carbonic anhydrase. So what does carbonic anhydrase do? It actually do two reactions. It can add oxygen to hemoglobin, as well as it can also control the formation of sodium bicarbonate, or you can say bicarbonate ions in the blood. So these are the two processes that carbonic anhydrase controls. We will discuss these in our future chapters in detail about what is carbonic anhydrase and how does it helps in blood pH and how does it helps in oxygen concentration. So it helps in oxygen binding with hemoglobin and also formation of bicarbonates in the blood. So these two processes are performed by a single enzyme, that is carbonic anhydrase. If we talk about what kind of enzyme it is, then you can say it is a non-regulatory enzyme. So non-regulatory enzymes follow induced fit model. So if we come to the conclusion, so which of these models is actually the one that enzyme follow, then the answer is both. Both of these models are followed by the enzymes. Like these, like the enzymes that are regulatory enzymes, they follow lock and key model, just like sucrase and maltase. Sucrase only act on sucrose, maltase only act on maltose, while non-regulatory enzymes follow induced fit model, just like carbonic anhydrase. Carbonic anhydrase can also add oxygen to hemoglobin, and it can also help in the formation of bicarbonate ion, that is H2CO3. So carbonic anhydrase is performing two different processes, but you need to know that non-regulatory enzymes only performs related processes. For example, if the enzyme is helping in digestion, then it will help in related digestion processes. It doesn't help in different processes. Like if an enzyme is doing digestion and in the another reaction the same enzyme is doing like nerve impulse, that is not possible. An enzyme can only form different reactions that are related to each other, just like carbonic anhydrase is only helping in the blood processes, right? It is balancing out bicarbonate ion in blood, and it is also helping in the attachment of oxygen with hemoglobin. Both of these processes are occurring in blood. Carbonic anhydrase is helping these two reactions that are closely related to each other. It's not that these enzymes act on completely different reactions, that is not possible. The non-regulatory enzymes only help in reactions that are closely related with each other. Let's move on to our next topic, that is about the enzyme substrate complex and catalytic mechanisms. So how does enzyme actually works? As you can see in the diagram, the enzyme and substrates attach with each other, and then the enzyme has to go from this position to this position. Why is that the position of enzyme is higher and lower? The reason is that before the reaction, the enzyme and substrates were on a higher energy level, and they reacted with each other and they used up some of their energy. That's why they lost it and they came below the graph. As you can see, when the product is formed, then the energy is lost. So as you can see, that means you need energy to for enzyme and substrate to react with each other. So when enzyme and substrate reacts, then you need a specific amount of energy so that enzyme and substrate could react and they can form products. So that energy is actually the activation energy, the energy that is used to form enzyme substrate complex is called as activation energy. We will discuss about activation energy in detail, like whatever we will discuss it now. Like just let's discuss about activation energy. So what is activation energy? Activation energy is the energy required to form enzyme substrate complex. So how do enzyme actually like lower the activation energy? Where do the energy that was used in the old reaction goes? For example, if there is no enzyme, then this is the pathway for substrate to convert into a product. You can see you will need a lot amount of energy, but with enzyme you are only needing small energy. So how does the enzyme actually decrease the energy? The enzyme does not do anything with the energy. It just only changes the path of reaction, just like we discussed about displacement in physics. For example, if you want to go from point A to point B, so point A is substrate and point B is product, and this is the pathway that you need to take to go to point B. But imagine enzyme gives you a shortcut, the displacement path, that is from A to B, and that's how you can save a lot amount of energy by directly going from point A to point B, and that's how activation energy is lowered at this point. So that's how enzyme works. How does enzyme lowers the activation energy? It actually gives new path to the reaction, so energy is saved and reaction is actually really becomes faster because of enzymes. So this was about enzyme substrate complex and the mechanism. If we talk about enzyme kinetics, then let's talk about the details. First of all, what is the Michaelis-Menten curve? Like first let's see about the graph that is in front of you. When substrate concentration is is formed in a graph between substrate concentration and reaction rate, the resulting curve is actually, as you can see, it is a hyperbola. This is a hyperbola. So why is it a hyperbola? At low substrate concentration, the rate increases steeply. As you can see, the reaction was increasing really fast, but as substrate concentration increased from a certain point, as you can see after this point, the graph became less steeper. It should have gone like this, but why is it going lower? Right, this is a question that is really important. The graph should have gone like that, but why is the graph going downward? The rate levels actually decreases. Why is that? Because at this point the substrate, the active site of the enzymes started becoming saturated. As you can see, at low substrate concentration, most enzyme active sites were empty. That's why the graph was really steep. When you were increasing substrate concentration, when you have one substrate, the reaction was this. When you had 5 substrate, then the reaction rate increased. When you had 10 substrate, then the reaction became really high. But as you can see, after 10 substrate, for example, I am taking it for approximation, after 10 substrates, the reaction didn't go as we planned. It started going less and less and less steep, and then it stopped increasing. What was the reason? The reason was because enzyme active sites started getting saturated, because you can increase substrate concentration as much as you want, but if you have a limited amount of enzymes, how can an enzyme substrate complex form? If enzyme substrate complex cannot form, then the reaction cannot increase, right? So after at intermediate point, some active sites were occupied and the rate was still increasing, but it was slow. But as you increase the substrate like to 100 or 200, then all the enzymes became occupied. You can't find more enzymes to react with the substrates. So after every enzyme molecule was used, then even if you increase substrate as much as you can, even if you have 1,000,000 substrates, the reaction will not go up, the reaction rate will not go up. So that's the beauty for this curve, that is telling you that even if you increase the substrate concentration, you also need more enzymes. As you have a limited amount of enzymes in your body, that's why the curve cannot increase infinitely. But if you have unlimited enzymes and unlimited substrates, then the graph will go like this. But as you have a limited amount of enzymes, limited amount of substrates, that's why the graph is actually a hyperbola. These are the definitions that are in front of you. You can memorize them, you can read them. We have just discussed all the points right now. Next thing is about the enzyme inhibition. Enzyme inhibition is really important concept because most of the students get these MCQs wrong. So first thing is about enzyme inhibition is that there are two types of enzyme inhibition. Enzyme inhibition is like a chemical substance. Inhibitor is a substance that can react with enzyme in place of substrate, but it is not transformed into a product, and thus it blocks the active site temporarily, or you can say permanently, and it is actually called as inhibitor, and the process is called enzyme inhibition. So the molecules which can act as inhibitors are poisons, antibiotics, cyanides, drugs, heavy metals. These are like examples of enzyme inhibitors. And in general, enzyme inhibition is normal part of enzyme activity. It does not occur like in an accident or a poison. It is a normal process that occurs in your body, because sometimes you need to stop an enzyme from working. That's why enzyme inhibition occurs. But there is another chances in which externally factors cause enzyme inhibition. For example, you get poisoned by a snake. The snake poison actually causes enzyme inhibition in your body, and the specific enzymes are stopped, and this is really dangerous for your life, because if enzymes stop working in your body, then all the reaction will stop and you will die eventually, right? So that's why enzymes are really important. And poison, what does the poison do? It acts as an enzyme inhibitor, and it stops the enzyme processes in your body. So there are types of inhibitor, the specific and non-specific enzyme inhibitors. If we talk about specific inhibitor, then they could be either be reversible or irreversible, and if they are reversible, then there are other types. It could be non-competitive, competitive, and non-competitive, that is on allosteric side. And if we talk about irreversible, then they are suicide inhibitors, they are time dependent inhibitors. If we talk about these enzymes in detail, then they are really important. First, if we talk about irreversible inhibitors. Irreversible inhibitor is just like its name is saying that the reaction is irreversible, right? So they occupy the active site by forming covalent bond or physically block the active site. What do? They forms covalent bonds. Like the covalent bonds, as you know, are strong bonds that can't be broken down really easily. So they form covalent bond with the active site. The example of irreversible inhibitors are cyanides and ions of heavy metals. Like cyanides are really potent poisons because they can kill an organism by inhibiting cytochrome oxidase. So what does cyanide do? Cyanide potentially block down, inhibit cytochrome oxidase. Cytochrome oxidase. So what is cytochrome oxidase? Cytochrome oxidase is an enzyme that is really important in your cellular respiration process, because cellular respiration is the process in which energy is made, ATP is made. If your body can't make energy, then you will eventually die. So cyanide actually block the cytochrome oxidase enzyme that helps in your cellular respiration process. If we talk about other, then the other example is heavy metals. Heavy metals are examples of irreversible reactions. So what does heavy metals do? Heavy metals actually combine with thiol group. Thiol group of enzymes. And what does it do? It actually breaks their disulfide bridges. What does they do? They break the disulfide bridges disulfide bridges of enzyme structure, and that's how enzyme structure is actually lost. You have studied about structure of proteins, and you know that enzyme is actually a tertiary or quaternary structure of proteins. Enzymes actually shows disulfide bridges, and it is present in tertiary and quaternary structures. So if the tertiary and quaternary structure of enzyme is lost, then enzyme cannot work, and heavy metals actually attach with the thiol group of enzyme, and they broke the disulfide bridges, and the enzyme structure would be destroyed. So that's how irreversible inhibitors acts on enzymes, and that's how they actually stops the working of enzyme. So the next thing that you need to know about irreversible reaction is that they are actually non-competitive. What are competitive and non-competitive inhibitors? They are actually the types of reversible reactions. What are reversible reactions? Reversible reactions are the reactions that could be reversed, right? In irreversible reaction, the damage was permanent, but in reversible inhibitors, the damage is not permanent, but it can be reversed. How we can reverse the process? It is simply by adding more substrates. If you add more substrates, then the reaction would be reversible and the inhibitor would be removed. For example, if we talk about competitive and non-competitive inhibition, first of all, let's talk about competitive reversible inhibition. So reversible has actually two types, competitive and non-competitive inhibition. First, if we talk about competitive inhibition, as you can see, it is a competition between substrate and an inhibitor. This is your enzyme right here, and there are two competitions. Either the substrate could come here, or either the inhibitor could come here. So there is a competition between substrate and inhibitor. If the inhibitor attaches with the enzyme, then the reaction would be blocked, but if the substrate attaches with the enzyme, then the reaction will continue. So how can we reversible the reaction? If we add more substrates, then you know that the chances of substrates attaching with the enzyme will increase, and the reaction would be reversed. Like if you have one inhibitor and if you have 100 substrates, then the inhibitor will have no choice but to lose, because the chances of inhibitor to attach with the enzyme is 1 out of 100. And if you keep increasing substrate, then the inhibitor will have less and less chances to attach with the enzyme. So that's our example of competitive inhibition. So competitive inhibition is nothing but a competition between substrate and a inhibitor. So this was example of competitive inhibitor. If we talk about the table, you know that active site binding is actually without inhibitor causes the substrate to bind, and inhibitor competes for the active site. And without inhibitor, the speed of reaction is maximum and Km is also maximum. But with competitive inhibitor, when the inhibitor attaches with the enzyme, then the rate of reaction does not change. Why is that? Because the reaction will not move forward, it will remain same, and the graph will stop at that point. If we talk about Km, the Km will actually increase because you will need higher amount of substrate. When you need higher amount of substrates, then Km will actually increase. So can we overcome this reaction? Yes, you can overcome it if we increase the quantity of substrates. If you keep increasing substrates, then the substrates will eventually displace the inhibitor, and it will take the position of inhibitor. That's why competitive inhibition is a competition between substrates and an inhibitor. So that was about competitive inhibition. If we talk about non-competitive inhibition, so non-competitive inhibition is the reaction in which there is no competition between substrate and the inhibitor. Why is that? Because the inhibitor does not actually attach with the active site. What does it mean? If I make the diagram here, as you can see, this is an enzyme, and this is the active site, and the substrate will attach on the active site. And here comes the inhibitor. Imagine this is an inhibitor. The in competitive inhibition, the inhibitor will go and compete with the substrate to attach on the active site. But in non-competitive inhibition, the inhibitor will attach any place other than the active site. So the place that is other than active site, as you know, is the allosteric site. So the inhibitor will attach on the allosteric site. So it will not actually compete with the substrate, but it will actually attach any place other than the active site. So that's how non-competitive inhibitor acts. So the non-competitive inhibitor actually attach with the allosteric site other than the active site. So how does this work? You need to know these tables. First of all, you need to know that competitive inhibition and non-competitive inhibition occurs in a different way. So as you can see in the competitive inhibitor, the inhibitor is actually combining with the active site of the enzyme, and in the non-competitive inhibitor it is attaching with the allosteric site of the enzyme. It is not attaching with the active site. If we talk about substrate binding, then in competitive inhibition the substrate would not be able to combine with the active site, but in non-competitive inhibition the substrate will attach with the active site just normally and chemical reaction. So how does non-competitive inhibition actually stop the chemical reaction? While the non-competitive inhibitor attaches with the allosteric site and it actually changes the structure of enzyme, it can change the bonding of the enzyme, it can destroy the structure, it can do anything with the enzyme that can stop the chemical reaction. So if we talk about competitive inhibition, it can be changed by increasing the number of substrates, but in non-competitive inhibition you cannot change by increasing the substrate concentration. You can increase enzyme structure, substrate concentration as much as you want. You cannot change the effect of non-competitive inhibition because the inhibitor is not attaching with the substrate, but it is actually attaching with the allosteric site. So if we talk about substrate binding, it is prevented, and in non-competitive inhibitor it is not, it is unaffected. If we talk about the Vmax, that is actually the speed of reaction, then it is actually unchanged in competitive inhibition. While if we talk about non-competitive inhibition, it is actually decreased. The rate of reaction is decreased. If we talk about Km, then Km is actually increased because the substrates actually number of substrates are increased, while Km remains the same in non-competitive inhibition because the substrate level does not change at all and it remains the same. While in competitive inhibitors the substrates are not attaching with the enzyme active site, that's why the Km will increase. The competitive inhibition is reversible with substrate, but non-competitive is not reversible with substrate. You cannot reverse the process by increasing substrates because the substrate are attaching with the active site, and the inhibitor is attaching to the place other than active site, that is allosteric site. Even if you increase the substrate concentration, nothing will change because inhibitor is not competing with the substrate at all. It is not competing with it. It is simply going to another place other than the active site. So this was all about the competitive inhibition and non-competitive inhibition. If we talk about the next step that is really important, that is factors affecting enzyme activity. So the first factor that affects the enzyme activity is temperature. If we increase the temperature, then obviously the enzyme reactivity will increase. If you keep increasing the temperature, the enzyme activity will keep increasing until it will reach a point that is called as the optimum temperature. The optimum temperature is the temperature at which the enzyme will work at its maximum capacity. If you increase the temperature more than optimum temperature, then it will actually be harmful for the enzyme. If we increase the temperature more than optimum temperature, what will it do? If you increase the temperature more than optimum temperature, what will happen? It will actually break the hydrogen bonds. So hydrogen bonds will break. So increasing too much temperature will destroy the hydrogen bonds in the enzymes, and that will cause the denaturation of the enzyme. Denaturation is called the process in which the enzyme structure is destroyed or lost when you increase the temperature more than it should be increased. So as you can see in the diagram here, if you increase the temperature kept increasing and increasing, and this is the point, this is the optimum temperature. At that point the enzyme activity actually became maximum, and after that point if you increase the temperature, the graph will go actually downward. The enzyme starts denaturating, and that's why the enzyme actually work at optimum temperature. The optimum temperature for human enzyme is actually 37 degrees Celsius. More than 37 degrees Celsius, and the enzymes in your body will start destroying itself, and it will cause denaturation of enzyme, and you will eventually die. The same is for the pH of the body. So enzyme has actually an optimum pH, and on that specific pH the enzyme works. If you increase or decrease the pH, then the enzyme activity will either increase or decrease. So at in specific pH, that is at which the enzyme will work at maximum quality or maximum efficiency, it is called as optimum pH. If you increase the pH more than the optimum pH, if you increase the pH more than the optimum pH, then what will happen? It will actually affect ionic bonds. So if you increase the pH, then the ionic bonds will be affected by increasing the pH more than the optimum pH. The pH, for example, in your stomach ranges from 1.5 to 3.5, that is the perfect condition for pepsin. If you increase the pH from 3.5 to 5, then the bonds will break in pepsin, and pepsin cannot work at that high pH. So that's how pH is really important. For example, in your blood, at the optimum pH is near 7.4, that is like physiological pH. There are some examples of pH. As you can see, the pepsin that is present in stomach works in acidic condition. Salivary amylase works in neutral environment. If we talk about pancreatic lipase that is present in your intestine, then the pH is actually basic. It works in alkaline pH. pH 8, trypsin and lysozyme, they both trypsin also works in alkaline pH, and lysozyme works in albeit slightly acidic situation. So that's how pH affects the enzyme reactions. If we talk about substrate concentration, at low substrate concentration, reaction rate is proportional to the substrates. Like if you keep increasing the substrate concentration, the reaction will keep increasing, and if you also increase the enzyme concentration, then obviously the reaction will keep increasing. This process can be easily shown in the graph. As you can see, if you keep increasing the substrate concentration like this, it will keep going up, and after a specific point it will not go up. Why is that? Because at that point the enzyme became saturated. You don't have enough enzymes to keep increasing the reaction rate of reaction. So if you keep increasing substrate and you also keep increasing enzyme, then you will get a slope like this. As you can see here, if the number of substrate and enzyme concentration keep increasing, then the rate of reaction will keep increasing like this, and you can go up to as much as you can. So these are the graphs in front of you for rate of reaction, for temperature, for pH, for substrate concentration and enzyme concentration. So if you increase temperature, enzyme activity will increase, pH also increase, substrate also increase, and enzyme concentration obviously it will increase. If we move on to our next topic, that is about cofactor, coenzyme and prosthetic groups. We already discussed them into our first of chapter, in the start of our chapter, so you can just revise it from here. I didn't knew that it was written here, so I just taught it in front of the chapter. So anyways, you can revise it again here. So enzyme contains two parts, protein and non-protein parts, and the non-protein parts would be cofactor or the Apoenzyme. Cofactor could be prosthetic group or coenzyme. Their details are written out here. We have already discussed all of it. If we talk about free energy and biological reaction and thermodynamics, so what is free energy? So energy, the change in free energy is actually determines whether the reaction is endothermic or endothermic, or it can be spontaneous or non-spontaneous. So these reactions are really important and they will help you out in chemistry section 2. So as you can see here, if we talk about the energy, when the substrate had the energy here and the reaction occurred, you needed an activation energy. After the activation energy, the energy of product decreased. So in this reaction, as you can see, energy was lost, right? The substrate used up energy and the product came at low energy level. So what type of process is this? Either it could be endothermic or it could be exothermic. So it is, as you can see, it is an exothermic process. So most of the enzyme reactions are actually exothermic. Why is that? Because you need activation energy for an enzyme to work. When you add an enzyme to a substrate, then the enzyme substrate complex will reach a point that is called as enzyme substrate complex, and that needs activation energy, and the remaining energy is actually called as the Gibbs free energy, and the free energy determines whether the reaction is endothermic or it is exothermic. As you can see in the diagram here, first the reaction was at a simple position like this, then the reactants combined with the enzyme and the graph went down like this and you got the products. In this reaction, the energy was lost, and that's why it is called as exothermic reaction. And in this reaction, as you can see, the reactant was at low energy, and after the reaction the products went to higher energy level, and in this reaction energy was absorbed. That is why it is called as endothermic reaction, or you can say endergonic reaction. So in exergonic reaction, energy is actually released from the system, and in endergonic reaction or endothermic reaction, energy is actually absorbed by the system. So the graph is in front of you. And if we talk about spontaneous and non-spontaneous processes, then exergonic reactions are spontaneous. Why is that? Because they don't need energy. As you can see, the reactants actually lose energy and the process happens on its own. It's just like you have a bucket of water like this, and you drop the water from your roof and the water will go down on its own. You don't need any energy to move the water from your roof. The water will move on its own from high point to the low point. And if we talk about the bucket, for example, if you have to take the water bucket from the ground floor to the roof, then obviously you have to take the water bucket using energy. You will lift the water and you will take it to the roof. That's why you need energy, and that is actually a non-spontaneous reaction. So spontaneous reactions are a reaction that happens on its own and they don't need any outside energy, and non-spontaneous reactions are the reactions that actually requires energy from the outside to happen. As you can see in the diagram here, the reactants needs energy to go from low point to a higher point. So that means the energy came from the outside. The reactants absorbed the energy and they were converted into products, and the products are at higher energy. That's why the endergonic or endothermic reactions are actually non-spontaneous. So that was all about spontaneous and non-spontaneous reactions. If we talk about the difference of energy here, as you can see, the reactants has more energy in exergonic reactions. The reactants are at higher energy and while products are at lower energy. If you talk about endergonic reactions, then the reactants are at low energy and products are at higher energy, because energy was absorbed during this process, and in exergonic reactions energy was released. So that is the process. The example of exergonic reaction is cellular respiration, and the process of example of endergonic is actually photosynthesis. In cellular respiration, energy is actually released, and in photosynthesis energy is actually absorbed. So these were the examples for exergonic and endergonic reactions. These reactions are actually really easy. If you try to understand them, then they are not really hard. Believe me, these topics are easy. Just try to take this lecture again and try to understand these topics again and again, and they would be really helpful in your chemistry section too. What are endergonic, what are exergonic, what are endothermic, and what are exothermic. If we talk about activation energy, we already discussed it. We are not going to discuss it again. You know that activation energy is the energy required to start or form energy substrate complex. It is the energy required to start a reaction. So how does enzyme lower the activation energy? Enzymes find a new path, or you can say enzymes use a displacement path, and energy is saved, and you will need less energy if you have enzyme, and without enzyme you will need to go through the real path, and the activation energy would be really high. So this was about the activation energy. So let's talk about some important tables. If you talk about competitive and non-competitive inhibition, these are the important tables. You can like discuss them again and again. We discussed everything in detail. Just read it out and you can take screenshot, but the notes would be available anyways. Memorize these tables and you are good to go. If you talk about endergonic and exergonic, this is your table again. And if you talk about coenzyme, cofactor, and prosthetic group, this is the difference between them. You can see that inorganic ions are metals, coenzyme are organic, and prosthetic could be organic or inorganic. So these are the differences between them, and this is the difference between log and key model. So these topics are really important. I didn't went into a really high detail for these topics. The reason was that because you don't need to go into a really deeper level, because enzyme chapter is important. Yes, it is important, but you need to know that where you have to stop for your IMAT preparation. You don't need to go into really high level of detail just to get 1 or 2 marks. You understanding my point? So getting this chapter was really, you can say, strategic. So these are the only things that you need to understand in this chapter. You don't need to like get anything else other than these topics. You are ready to go for this chapter. Like you have covered everything that there is for related to enzymes, and the enzyme chapter has been finally ended. And that's all for enzyme section. Do solve your don't forget to solve your practice MCQs, solve all your mock exams, and that's all from my side. Take care. Bye bye.